Dear viewers, it is my privilege to provide to you an introduction to Parkinson's disease in this series of ENIGMA-U lectures. My name is Isbrand van der Werf from Amsterdam in the Netherlands, and I'm the chair of the ENIGMA Parkinson's disease working group. So let's dive right into it. When we talk about the epidemiology of Parkinson's disease, it's important to know that it affects between 1 and 2 out of every 1,000 people in the world. More so in males than in females. It affects more people as they age. So over the age of 60, the prevalence would be 1%. It is at this moment the second most prevalent neurodegenerative disease after Alzheimer's disease, but it is also the fastest growing neurodegenerative disease in the world. For a limited number of cases, there are identifiable genetic factors that contribute to the disease. But we also know that there are environmental risk factors. And being such a prevalent disease, we probably all know cases of people who suffer from Parkinson's disease or have done so in the past. And you see a number of those people in the pictures below. And when we talk about the symptomatology of Parkinson's disease, it is primarily recognized as a motor disease, or at least traditionally it has been viewed as a motor disease. So the hallmarks of the diagnosis of Parkinson's is a slowing of movement, also known as bradykinesia, an absence of movement, which is hypokinesia, rigidity of movement, and then postural imbalance, meaning that these people have a tendency to fall easily and have problems with their posture. And in some, but not all cases, there is also a very noticeable tremor. And the characteristic posture of a Parkinson's disease patient is shown on the right. Somebody with a stooped posture leaning forward, a noted lack of movement in the arms, and a shuffling gait with small steps that they take. We now know that although the motor symptoms are very characteristic, there are, in fact, a lot of non-motor symptoms that also characterize the disease, and sometimes they do so already before the onset of the motor symptoms, and they range from autonomic dysfunction to a lack of smell, fatigue, pain, sleep disturbances, mood disorders, anxiety, impulse control disturbances, cognitive disturbances, sometimes even leading to a full-blown dementia, and psychosis and hallucinations. So this is really a disease that affects many aspects of life and can be very debilitating for the sufferers. It appears now that the non-motor symptoms that are listed here are in fact more detrimental to the quality of life as compared to the motor symptoms. That is, that the patients report that these non-motor symptoms affect them much more than the possible motor symptoms that we know so well. And therefore, it's also of importance to keep those in mind when we talk about Parkinson's disease. And so, like I said, the hallmark of Parkinson's disease, and at least for the diagnosis of Parkinson's disease, is the very obvious motor impairments. And these stem primarily from a degeneration of the dopaminergic system in the midbrain that we see here on the left. So in the image on the left, we see a slice through the brain at the level of the mesencephalon, the midbrain. And on the left of that image, we see a Parkinson's disease brain, where we see a noted absence of that black staining that we see on the right side of the image. Indicating that the dopaminergic system in the substantia nigra has degenerated, and therefore there's a lack of dopaminergic cells in the brains of people who suffer from Parkinson's. At a more whole brain level, we know that the diagnosis of Parkinson's can be made by looking at the Lewy bodies and the alpha-synuclein pathology. And so what we see on the right side of the slide here are the things stained in the dark color, in black and brown, and those are either intracellular inclusions of protein, alpha-synuclein, that we see here in image B, indicating that these cells are full of this protein, misfolded protein. And we also see on the in figure A what we call Lewy neurites, and they are also stained for alpha-synuclein. So the deposition, intercellular deposition inside the neurons of alpha-synuclein is in fact the post-mortem pathological hallmark of the disease. And the proper diagnosis of Parkinson's can in fact only be made post-mortem. And using post-mortem material, already many years ago, colleagues from Germany by the name of Braak have described that there's a progression of the alpha-synuclein pathology in the brain. And Braak distinguished 6 stages of the disease with progressing alpha-synuclein deposition. And across those 6 disease stages, there is an orderly progression of the alpha-synuclein staining inside the brain, typically starting in the brain stem, as we can see here in the image, and then moving upwards towards the midbrain, and then from the midbrain, eventually also reaching the cortex until great parts of the cortex have also been affected. Note that there's here on the ventral side of the frontal lobe, there is the olfactory bulb, which is also one of the areas that very early on in the disease shows the hallmarks of the alpha-synuclein pathology. And so those are the regions that we're looking at, typically starting then in the olfactory bulb and also in the brainstem, and then moving up. And it is only when the pathology hits the midbrain here that we start seeing the motor disorders, which means that the pathological process has already been ongoing for a number of years prior to the onset of the slowing of movement, the absence of movement, and potentially the tremor. It also means that many other functions, apart from the motor disorders, are involved in Parkinson's disease, those that have to do with the brainstem and also those that have to do in later stages with the cortical atrophy. And this is in fact something that we have noticed when we look at MRI images of the brain in people who suffer from Parkinson's disease. And these stages that are mentioned here are stages following the diagnosis of Parkinson's. So stage 1 would be a newly diagnosed individual who has just shown the motor symptoms, and we see that at this stage there is very limited involvement of the cortex. According to the theory of Braak, this is because the pathology is at this moment still in the brain stem primarily. And then we see as the disease progresses across stages 2, 3, and then 4 and 5, which are the final stages, that we progressively see involvement of more cortical areas. And you can imagine that the atrophy that we see in those cortical areas also contribute to other kinds of disorders like cognitive and emotional disorders. And so when we go back to the dopaminergic system that we see here in a healthy brain at the level of the mesencephalon, again, we see the black staining, which indicates the presence of the dopaminergic cells in the mesencephalon in the substantia nigra. And it's important to realize that these dopaminergic cells project to a number of regions in the brain, as we see on the right. There is a strong projection to areas that we call the striatum, and then there are other projections that go towards dispersed areas of the cortex. And again, this lack of dopamine that arises as part of the Parkinson's disease process affect multiple areas of the brain directly through the lack of dopamine, and then later on also indirectly through the atrophy. And so when we take a closer look at the substantia nigra, we see here the level of the mesencephalon. We recognize the cerebral aqueduct here, and then we see here the cerebral peduncles of the brain. And then on the inside of the cerebral peduncle, we see that very conspicuous black lining, which would be the substantia nigra pars compacta, which is the location of the dopaminergic cell bodies. And the presence of this black staining here indicates that we are looking at a non-Parkinsonian brain. And like I said, one of the important targets of the dopaminergic system is the striatum. And the basal ganglia are important structures in the brain that are involved in movement, preparation of movement, and we see them here in the red coloring. And in the next slides, we will talk a little bit about how those basal ganglia structures affect movement, but also other functions of the brain. Here on the left, we see another rendition of the basal ganglia, we see that it's a very complex structure, very much a 3D structure. We see areas that we know are called the caudate nucleus, this elongated structure here. And then more centrally, we see the putamen. And then towards the midline, there is also areas that we call the globus pallidus. Now, in a cross-section of the brain, we see those same structures here, and I will try to indicate them here. We see the caudate nucleus lying lateral to the lateral ventricle. We see here the putamen, and then on the inside of the putamen, we see the globus pallidus, the external part, and here the internal part. For reference, we see the thalamus here located on the midline, both sides, of course, on both sides of the third ventricle of the brain. And if we put that in schema, then the flow of information that is involved in motor preparation comes from the cortex. So there is a stream of information from the cortex which goes through the internal capsule all the way down to the spinal cord to initiate movement. And we see that surrounding that descending projection down to the spinal cord, we have the structures of the basal ganglia indicated here. So in red is the striatum, consisting of the caudate nucleus, the putamen, and a specialized structure called the nucleus accumbens. In red, we see the globus pallidus pars externa, the pars interna, the ventral pallidum here, and here we see then structures like the substantia nigra pars compacta that are involved in the functioning of the basal ganglia. And I will take you step by step through the involvement of these structures and the influence that the dopaminergic degeneration has on its function. And so when we look at the normal situation, it is important to realize that there are two pathways that flow through the basal ganglia in order to modulate movement. So from the cortex, there is a projection into the striatum that we see here in red. And then we see from the striatum a projection into the internal segment of the globus pallidus, which then leads to a projection to the thalamus, and the thalamus then communicates back to the cortex in order to start the movement process. So the projection down into the spinal cord, indicated in gray here, happens only after the processing through the basal ganglia. Now notice that this is what we call the direct pathway. And this will become clear in a moment when we talk about the other pathway, which is the indirect pathway. So the direct pathway goes from striatum to the internal segment of the globus pallidus to the thalamus. Notice that we are looking at excitatory transmission here at the level of striatum, an inhibitory transmission here towards the internal segment of the globus pallidus, and another inhibitory projection to the thalamus, meaning that we have a double inhibitory transmission, which then leads to a net excitation, right? So double inhibition means excitation. You can think of it as disinhibition. The other pathway is what we call the indirect pathway, and it follows more or less the same route. So again, the first projection is from the cortex into the striatum, indicated in red. And then there is a few more stops along the way. So from the striatum into the external segment of the globus pallidus, into the subthalamic nucleus, indicated in green, from the subthalamic back into the internal segment of the globus pallidus, and then it follows the same route from the globus pallidus interna again to the thalamus. Now notice that in this particular projection, we have three inhibitory projections, one here at the level of the external segment of the globus pallidus, one here at the level of the subthalamic, and then again here, one at the level of internal segment of the globus pallidus towards the thalamus. And therefore, three inhibitory projections in a row will lead to a net inhibition, which means that we have a direct pathway, which in principle is excitatory, it promotes movement, and an indirect pathway, which inhibits movement. And those two pathways together organize the output of movement from the cortex and its projection down into the spinal cord. Now, important to realize is that the substantia nigra pars compacta, here indicated in yellow, sends a projection of dopamine to both the inhibitory and the excitatory pathway. What happens is that dopamine here at the level of the striatum inhibits the indirect pathway, but excites the direct pathway. And that means that in the normal situation, in principle, movement is permitted, is allowed, because there's a slight preference of the direct pathway. And you can imagine what happens when the dopamine disappears. That preference for movement permission is reduced, which leads to a net inhibition of movement, and therefore the lack of movement, the slowness of movement that we see as part of Parkinson's. Right, and that is what we see here in the Parkinsonian state, where we see a disruption of the dopaminergic inhibition, leading to a slight strengthening of the indirect pathway, which means that as a net result, we have a reduction of movement in the Parkinsonian state. This is how we think about the movement disorders as part of Parkinson's. But like I said, many more things happen in the Parkinson's disease process. And one of them is that as the cortical targets also get affected, there are also cognitive and affective disorders that we see. And it would appear that in Parkinson's disease, across the disease duration, there is a high probability of people also developing dementia, and we call this Parkinson's disease dementia, because it has a few qualities that are maybe different from the dementia that we see as part of other diseases, typically Alzheimer's. And we see that given enough time, many people suffering from Alzheimer's will in fact develop a degree of cognitive disorders, and in some cases actually leading to a full-blown dementia. And that is indicated also in this slightly complicated graph that I encourage you to look up from the original paper by Dag Arsland, published in Nature, which indicates that over time, there are different trajectories that people can follow. Some people from the early stages, when they already have minimal cognitive impairment, go into a rapid progression towards Parkinson's disease dementia. Other people have a slower trajectory towards Parkinson's disease dementia, and some people only maintain minimal cognitive impairment. And then there is a small group of people that show no or minimal cognitive decline across the disease process. And so there is a wide variety in the population of people with Parkinson's disease with respect to their cognitive abilities. And this is also what we see when we look in the ENIGMA Parkinsons working group data, that the association of cortical thinning, so the cortical atrophy that we see specifically in relation to cognition, is part of the general atrophy that I showed you before. So these are the areas that are associated with cognitive decline in Parkinson's. And you see that the cortical areas are co-segregating, so they're correlating with the increase of cognitive decline. At the cortical level, but also at the subcortical level, we see that there are decreases in volume in various parts of the basal ganglia that we talked about in relation to cognitive decline. One important aspect of Parkinson's disease is something that we know since about 10 years, and that is that one of the earliest symptoms already predicting the onset of Parkinson's disease and preceding the motor symptoms with maybe 5 years or 10 years and sometimes even longer than that is a very specific sleep disorder. So the thing that we call REM sleep disorder or REM sleep behavior disorder, RBD, for short, is a symptom in people with Parkinson's disease who, in people who may develop Parkinson's disease, where they show a lack of the typical atonia during REM sleep. So in the normal situation, people undergoing REM sleep are paralyzed from the neck down. They do not move their limbs, their arms, their feet, and their legs. People who have RBD, in some cases, will show movements during REM sleep. And this means that they will be acting out the contents of their dreams. And so when they are dreaming about moving and particular movements, they will actually show those movements, whereas in the normal situation, you would be lying perfectly still. And in some cases, this may mean that the feet are moving, the legs are moving, if the person is dreaming about running or walking. Sometimes those dream images are violent and could involve fighting or warding off aggressors, and then you may actually see people fighting in their REM sleep. And the reason that this is important is that this is a very reliable marker in people who have not yet developed full-blown Parkinson's disease, so they do not have the motor symptoms, but they do already have something that is called RBD. And it is important to understand that not everybody has RBD predicting their Parkinson's disease. But when you do have RBD, and you can exclude other causes for RBD, which makes it then what we call idiopathic RBD, that is RBD that cannot be explained on the basis of any other cause, that these people will, with a high probability, develop Parkinson's disease or a related disorder, alpha-synuclein related disorder in the future. And it's shown here that if you start with the group of people diagnosed with idiopathic RBD, that given enough time, 5 years, 10 years, and sometimes 15 years down the road, almost all these people will have converted to an alpha-synuclein related disease. Typically Parkinson's disease, but also the related disorder, dementia with Lewy bodies. And so we have here a very early symptom that has been recognized over the last 10 years as being very predictive of a Parkinsonism. And that is, of course, something of great interest, because that means that perhaps we have a window of time in which the motor symptoms have not yet developed, and perhaps we will be able, given enough research and technological advances, to maybe halt or slow the disease process before it reaches the midbrain and before it starts producing the characteristic motor disorders. Parkinson's disease is very much a field that is moving at a great pace. We are learning much more about Parkinson's disease as we go. And so researchers think that there are maybe variants of Parkinson's disease, some variants that are maybe coming from the body, and then from the body into the brain, developing then Parkinsonian symptoms, and that there are other variants of Parkinson's disease that really start within the brain itself. And this is very much ongoing work, and so this is something that would be of interest in the coming years to see if we're looking at one disease or whether we are looking at a disease that has multiple roots and multiple causes and multiple trajectories. And it is also for this reason, of course, that we have chosen to study Parkinson's disease in this worldwide initiative of ENIGMA. This is a very short introduction to Parkinson's disease. I very much hope that it encourages you to go out and look for relevant literature, and we hope to see you in other instances of the ENIGMA-U lectures. Thank you very much.